Hi students, welcome to the next session in oral pathology. We are discussing odontogenic tumors, and in this video we are going to discuss about CEOT, calcifying epithelial odontogenic tumor. Right, CEOT, calcifying epithelial odontogenic tumor, or the other name of this tumor is your Pinborg tumor. Okay, Pinborg tumor is the other name by the scientist. Okay, so what to learn in CEOT? What is the importance of CEOT? So far we have learned that most of the odontogenic tumors, they arise from your dental lamina or its remnants like the rest of cirri. Okay, so the origin of CEOT is little interesting. What about the cell of origin from where this is originating? Okay, the COC originating, obviously it originates from the rest of dental lamina, but the first important area of its occurrence is considered to be stratum intermedium. Okay, when you have learned about the development of a tooth bud in your dental histology, you may have heard about stratum intermedium. Means the inner enamel epithelium, outer enamel epithelium, and a layer of cells which is called stratum intermedium, you see in the advanced bell stage and all. Okay, those cells, those stratum intermedium cells is supposed to be the cause for this CEOT. If stratum intermedium is there in the ocean, you should go for stratum intermedium. The second best option is your dental lamina. Okay, dental lamina. The first best option for the origin of CEOT is considered to be stratum intermedium. Okay, so that is about your stratum, your CEOT's origin. Now, if you see a little about its clinical features. Okay, now you know that AOT favors to come in the anterior maxilla, but CEOT usually favors your posterior mandible. Okay, posterior mandible is the favorite side. Most common side we say is a posterior mandible, and usually it comes along with an impacted tooth. Impacted tooth association is there. Okay, an impacted tooth association is there, and most commonly COT comes in the posterior mandible. And what about the radiograph? Can you see this picture? I want to show you this picture. Okay, can you see this radiographic picture? What you see here is that you can see an impacted tooth here, and you can see a radiolucency here. Okay, I can appreciate a radiolucency there and an impacted tooth there. And you know, from the name itself, now you can guess some things. Calcifying epithelial odontogenic tumor. So there is some kind of calcifications in this, right? Some kind of you can expect some kind of calcification. When we discussed about what our calcifying odontogenic cyst. Okay, we discussed about calcifying odontogenic cyst, and there was also some kind of calcification in the lining, right? Those ghost cells get calcified. So similarly, CEOT, from the name CEOT, we can expect that there is some kind of calcifications in this, right? That is our common sense. Now you see this along with this impacted tooth, I can see some kinds of streaks there, right? I can see some streaks of radio opacity there. Can you see that? Okay, so that is some kind of calcifications. Okay, some kind of calcification. So radiograph here will be a radiolucent, well unilocular or a unilocular, well what bordered, smooth bordered radiolucency with the radio opacities inside. Okay, so it is having radio opacity. Some kind of radio opacities are seen within this radiolucency. So about the radiograph, if you see radiology feature, you can see that there is well defined radiolucency. Defined radiolucency is there, and there is a smooth radio opacities. Okay, radio opacities. That smooth radio opacities have a particular name that is called a snow driven pattern. Snow driven pattern. Snow driven. Have you seen driven snow? What do you mean by driven snow? Okay, when in the mountains, when the first snowfall is happening, so one layer of snow is formed, above that other layer of snow is formed, and when it goes undisturbed, it will have a very smooth pattern of deposition, right? The snow will fall in a very smooth pattern, and when it goes undisturbed, it will be very smooth layers. Okay, so that is driven snow pattern. It will appear very smooth in layers. Okay, so driven snow pattern or snow driven pattern, that type of radiolucency radiograph is seen in CEOT. So that is a radiolucency with this scattered smooth radio opacities. Okay, so that's about your radiological feature. Now coming to histology, what to learn in the histopathology of your CEOT? What to learn? CEOT, the cells of CEOTs are very, very particular. Calcifying epithelial odontogenic tumor. In calcifying epithelial odontogenic tumor, we see polygonal cells. Okay, we see polygonal cells. And what's the peculiarity of this polygonal cells is that they are very close to each other, very attached to each other. Okay, you can see that this polygonal cells, they are very attached, and you can see that the attachment is very strong. You can see the prominent intercellular bridges. Cell to cell attachment is through intercellular bridges, right? A cell attached to the next cell through intercellular bridges. And in CEOT, what striking feature is that you can see polygonal cells. Okay, we can see polygonal cells with a prominent, very prominent intercellular bridges. Intercellular bridges. Okay, means they are very much bonded to each other, very much connected to each other. Okay, that is one important feature of CEOT. And along with the CEOT, along with these intercellular bridges, outside the cell, you can see some kind of pink structures, some pink deposits outside the cell, extracellular. Okay, not inside the cell, outside the cell, you can see some kind of extracellular eosinophilic or pink colored pink deposits. And what are these deposits? What are these? They are amyloid. So amyloid deposits are seen, pink colored deposits. Okay, and these amyloid deposits, by time, what happens to them? By time they get calcified. Okay, they get calcified, and they calcify in a pattern. They will get calcified, and you can see this type of calcification. Okay, when they get calcified, they turn blue in color. Okay, calcified materials in the HNE, hematoxylin and eosin stain, when they get calcified, they turns bluish in color. Okay, blue, it takes more of hematox, calcified materials. And these calcified materials, which look like a ring like calcification or lamellated calcification, we say lamellated. Lamellated means ring like. Okay, one lamellae, just like lamellae. Okay, so these are called your Lees-Gang rings. Okay, Lees-Gang rings. Now, very important, Lees-Gang rings are not only seen in CEOT, we have seen that it is already also seen in AOT also. Okay, so Lees-Gang rings are seen both in CEOT and AOT, more prominently in CEOT. Okay, if they ask you to select which among the following shows Lees-Gang rings, and if you are asked to select only one option, you select CEOT. Okay, please note, if you are asked to select one, you should select CEOT. But you should see whether both AOT and CEOT is there in the option. Okay, if there is any option that both AOT and CEOT, then you go for both, because in AOT also you see a particular type, that is your CEOT like areas. Okay, CEOT like areas, those areas show Lees-Gang rings, right? So this is about your CEOT. Okay, so I will show you the image. And here is a histological image. Okay, in this picture you can see lot of Lees-Gang rings. Can you see this? Okay, they are all lot of Lees-Gang rings. Okay, see the bluish ring like structures, and the cells here, these cells are polygonal cells. Okay, polygonal cells with a prominent intercellular bridge. I think this is more clear in this picture. Okay, it is more clear in this picture, and wow, it is very nice. You can see so much of polygonal cells with the prominent bridges, and you can see this amyloid like material. See the amyloid like material. Okay, amyloid like material is here, and some places it is calcified. When it is calcified, it changes into blue in color. Okay, rest all these are all amyloid like material. Can you see this? These are all amyloid like material in that, and when it gets calcified, it turns little bluish. Okay, so this is a higher power view where you can see this polygonal cells. I think these are all polygonal cells here, and they have a very good intercellular connections. Okay, you can see if I have magnified it maximum, you can see that the cells are very much polygonal. They are very much polygonal in shape, and see the intercellular bridges there, they are very connected to each other. Okay, so this is how a CEOT is seen. Okay, CEOT. Now there was a question in your NEET INICT paper. How can you identify that this is an amyloid? Those material are amyloid. So that is it. How to diagnose CEOT? How to diagnose CEOT? How can we diagnose CEOT? How to know that that material is amyloid? So there is a few methods. Okay, so you take a slide. You take a slide of CEOT. I have taken a slide of CEOT. Okay. So first, what I am do, I am doing an HNE stain. I do an HE stain, hematoxylin and eosin HE stain. That is a normal stain. Now, what I will see that those materials are pink in color. What is that material showing? Material will show up as pink in color. They will take the eosin, and they will be taking eosin, and that will be pink in color. Okay, done. Now I have to see whether this pink colored material is amyloid or not. So what I do is that I take another slide. Okay, I take another slide of CEOT. What I am going to do is I am going to stain with thioflavin T. Thioflavin T. So this time I have changed the stain. Okay, thioflavin T. And what I am going to do is that I am going to see this side in a fluorescent microscope. In a fluorescent microscope. So that is a particular microscope, fluorescent microscope, and I can see that these material which were pink in the HNE now starts showing strong blue what fluorescence. Okay, it will show strong blue color fluorescence. Okay, it will show strong fluorescence, right? Thioflavin T. Now I'm okay with it. Now let me see whether it is amyloid, but I don't have a fluorescent microscope. Fluorescent microscope is very expensive, so I don't have a fluorescent microscope. So I am thinking, what other method I can do? I take CEOT slide and I stain with a Congo red. I stain with a stain called a Congo red. Okay, so I use this Congo red, and what I did that I used a polarizing microscope. Okay, polarizing microscope. Now polarizing microscope is less expensive. It is cheaper, and we can attach this polarizer to any microscope. So I can't afford a fluorescent microscope. I am using a polarizing microscope, but this time I stained with a Congo red. Now what I can see is that when I see this under a polarizing microscope, I can see this material as apple green color. Apple green by refringence. It will show apple green by refringence. It's the optical property. Okay, optical property of showing refringence by refringence. It will give apple green color. Can you see this? I will show the image. Oh, see this. See. Okay, those pink materials which was dispersed between the cells. Okay, those pink materials which were dispersed between the cells, when I stained with Congo red and I observed it in a polarizing microscope, I could see this green color, this apple green color. Okay, you may have seen green apples, right? Apple green birefringence. It's the optical property of a polarized light to show up a apple green birefringence. Okay, so this is how we can diagnose a CEOT. Okay, so I hope you understood CEOT. Okay, these are certain important areas what they ask for. Okay. So see you in the next video with the next topic. Thank you.